So in viruses, we're going to be renaming the viral messenger RNA, unfortunately, because we like to rename things in biology. So viral messenger RNA is no longer called that. It is going to be called plus single-stranded RNA. And you'll see why we end up renaming the viral messenger RNA as plus single-stranded RNA, and why we're going to be renaming all of this other nucleic acid as well as we go along with the viral life cycles, because viruses are a little bit strange with what they're using for their genetic material. So anyway, here is our viral messenger RNA, or plus single-stranded RNA. Our example here, we've got the 5' to 3', and here's our sequence, GAC, etc. here. And down here we've just rewritten our plus single-stranded RNA, which is our viral messenger RNA, and same sequence running 5' to 3'. Okay, if we have a minus or negative single-stranded RNA, what that means is that it's going to be anti-parallel, so running the opposite direction. So you can see that it's going to be running 3' to 5' instead of 5' to 3' as the plus strand, and it's also going to be complementary, which just means that the G's and C's, A's and U's will pair up there. So the minus strand is going to be complementary to the plus single-stranded RNA. So it'll be anti-parallel and complementary to the plus strand. So the minus strand is going to be anti-parallel and complementary to the plus strand. Okay, and then if you have plus single strand of DNA, that just means that it's going to be going the same direction. So it's going to be running 5' to 3' as the plus single-stranded RNA, and it's going to have the same sequence. As the plus single-stranded RNA. Okay, so if we look at our comparison here with our plus single-stranded DNA, we've got a G, and here's our plus single-stranded RNA, we've got a G there, A's and A's, C's and C's. Now the only difference is we have T's in DNA instead of U's, because remember, in DNA we have T's, and RNA we have U's instead, so that's the only difference there. Okay, and then if we have a minus or negative strand of DNA, a single strand of that, that's going to be anti-parallel and complementary to the plus single-stranded RNA. So if we look at our minus strand, it's going to be running 3' to 5' compared to our plus single-stranded RNA running 5' to 3', and it's going to be complementary, so G's and C's, A's and T's will match up. And remember, with the DNA we have T's instead of U's there. Okay, so what if we have double-stranded DNA? So the two strands of DNA, remember, are going to be anti-parallel and complementary to each other. So double-stranded DNA just means that you're going to have a plus single strand of DNA and a minus single strand of DNA. So the plus strand would be running 5 to 3 prime. And then the minus single strand of DNA would be running the opposite direction, 3 to 5 prime. And they're going to be complementary to each other. So double-stranded DNA, it just consists of a plus single strand of DNA and a minus single strand of DNA just joined together. That would be a double strand of DNA. Okay, so just important to remember that viral DNA is no longer called that. So viral DNA, viral messenger RNA is now called plus single-stranded RNA. So that's what we're now going to call it. Okay, and we only do this in viruses. This is the only one we do it in. We don't do this in cells, bacteria, nothing like that. So just in viruses, we rename it like this. Okay, so let's take a look at our group 1 viruses. They're going to be using double-stranded DNA as their genetic material. And I've just given you some examples of some group 1 viruses. There's some bacteriophages here that I've given you examples of, and also some eukaryotic viruses. You do not have to memorize which viruses are in group 1. And because these viruses are using double-stranded DNA as their genetic material, cells also use double-stranded DNA as their genetic material. So this is going to look very similar to the way that cells are, using their double-stranded DNA. And a lot of these viruses will rely on the host cell's DNA and RNA polymerases. So they don't have to bring a lot of their own enzymes with them because they can just use the host cells. All right, so let's go ahead and we'll draw out the life cycle for our group 1 viruses here. And I'll show you what you need to know. So when we make a virus, we only need to make 2 things. We just need to make more of the viral nucleic acid. So when our virus infects our host cell, we've got our viral nucleic acid, which is double-stranded DNA. So we just need to make more of that. And we need to make the viral proteins to make our capsid. So those are the only 2 things we need to make. Okay, so when our virus infects our host cell, we've got our double-stranded DNA here from the virus, and it's going to direct how to make more virus. And so our cell looks at this and it says, oh, I recognize this. This is like what we use in our cells. Cells use double-stranded DNA as their genetic material. And so the cell says, okay, I will go ahead and transcribe this for you. And we'll make the messenger RNA, which remember in viruses, we've changed the name and we're going to call the messenger RNA plus single-stranded RNA. And then the host cell ribosomes recognize the messenger RNA or the plus single-stranded RNA, and they will go ahead and translate that and make the viral proteins. And then these viral proteins will self-assemble and they will make the viral capsid. Okay, so that's one thing done. We've made our viral capsid, and then the other thing we need to do is we just need to make more of our viral double-stranded DNA, the nucleic acid it's using, and it's its genetic material. So all we're going to do is we're just going to do DNA replication. And then we can just put that viral nucleic acid inside the capsid. And there's our newly made virus. Okay, so now we're just going to fill in the names of the enzymes. So the enzyme that's used to do transcription, remember we're going to use RNA polymerase to do transcription because RNA polymerase makes RNA. But in viruses, we're going to add a little bit more information to the name. So we want to look back and say, what are we using as the template? So we're using DNA as our template. That's what we're transcribing to make this RNA. So we're going to call this a DNA-dependent RNA polymerase. Okay, so that would be the name of that enzyme. And then the enzyme that we're going to use to do DNA replication here, remember that DNA polymerase makes DNA. And again, with viruses, we're going to add a little bit more to this name. And so we're going to look back and say, well, what are we using as our template to make this DNA here? And so our template is DNA. So this would be a DNA-dependent DNA polymerase. Okay, so what I would do for the test is I would put some blanks. So I might put a blank here and say, well, what's the process that's going on here? And you would have to fill in transcription. Or this might be blank, where you'd have to fill in, oh, the process going on there is translation. Or maybe I'd put a blank here and say, well, what product is being made here? Well, that would be the plus single-stranded RNA. Or maybe I put a blank here and say, well, what's being made here? What's this product? And this one's easy because whatever nucleic acid is being put into the capsid, it has to match what you started with. Whatever the nucleic acid is that you start with, that has to match whatever goes into your capsid. Okay, so these two have to match. So that has to be double-stranded DNA. Or I might put a blank here and say, well, what's the enzyme that's being used here? And that would be the DNA-dependent RNA polymerase that's doing transcription. And you'd have to give the full name. If you just say RNA polymerase, you'd only give half credit because you need to say it's DNA-dependent RNA polymerase. Okay, so those would be the kind of questions that I could ask about that. And there's a homework question on the Chapter 6 homework that gives you an idea of what I would ask as far as a test question on this. And so you're not going to have to draw out this whole picture. I would draw this out, but I would just leave some blanks on it.